Okay, hello to all of you. Let's discuss chapter number 9, part 2. So, in the second part, we are going to talk about fermentation basically, and let me go to this short note. So, fermentation, as I have already said, that it's an anaerobic process and it's a partial degradation of the sugars, and it produces only two molecules of ATP, but it is quicker. So, So fermentation is basically glycolysis. So we have already discussed in the process of aerobic respiration or cellular respiration what glycolysis is. So basically fermentation is glycolysis with one extra step. Okay? So this is a way of extracting energy, but it involves a partial degradation of the sugars. So there are two kinds of fermentations. One is alcoholic fermentation, other is lactic acid fermentation. So you will be doing a lab also on fermentation and also on photosynthesis. Photosynthesis we're going to discuss in chapter number 10. So basically in alcoholic fermentation, so glucose by the process of glycolysis gets converted into pyruvate. And then this pyruvate, which has three carbons, it loses carbon dioxide, okay, and then it gets converted into acetaldehyde. Okay. And then this acetaldehyde that gets converted into ethanol. So pyruvate loses carbon dioxide, which means it loses one carbon, and the compound which is formed is acetaldehyde, and this acetaldehyde then gets converted to ethanol. And what is happening here is acetaldehyde is actually getting reduced. So acetaldehyde is actually gaining electrons and it's getting converted into ethanol. So from where these electrons are coming? So if you remember, in glycolysis, two molecules of NADH are produced. So two NAD plus, they get converted into NADH. So two NAD plus, they get reduced, right, and they get converted into NADH. And NADH, in the process of glycolysis, in the process of cellular respiration, it takes these electrons to the inner membrane of mitochondria, and there they are delivered to the inner membrane of mitochondria, and then these electrons flow as we all discussed, right? And eventually they're taken by oxygen. And in the aerobic respiration or cellular respiration, oxygen acts as the final electron acceptor. So in the case of fermentation, basically, as it's an anaerobic process, oxygen is not involved. But in this case, an intermediate metabolite, like here acetaldehyde, it is actually acting as the final electron acceptor. So the electrons which were taken by NAD plus and NAD plus was converted into NADH, those electrons, they are accepted by acetaldehyde, and acetaldehyde is converted into ethanol. So acetaldehyde gets reduced and ethanol is formed, and NADH gets oxidized. It gets oxidized. So NAD plus gets reduced to NADH in glycolysis, and now in this step NADH delivers the electrons to acetaldehyde. Acetaldehyde gets reduced because it accepts the electrons. It gets converted into ethanol, and NADH gets oxidized to NAD plus. So this is alcoholic fermentation. So the typical example of alcoholic fermentation is when we make the bread at home. So usually we take the yeast, we activate it, and we add into the all-purpose flour. And after some time, you can see the flour starts rising, and that rising is because of carbon dioxide. So that is a typical example of alcoholic fermentation. So in this process, when we are making bread or these things, so in this process, a little bit of alcohol is also formed, but it gets evaporated during the baking process. Alcoholic fermentation is basically used in the process of wine making and beers and all the alcoholic beverages. The other kind of fermentation is lactic acid fermentation. So it's basically the same. So it involves glycolysis. Glucose gets converted into pyruvate. Then pyruvate is converted into lactate. In this thing, in glycolysis, when glucose is converted into pyruvate, NAD plus gets converted into NADH, okay, and then this pyruvate accepts the electrons. Pyruvate accepts the electrons from NADH. It accepts the electrons from NADH, and it gets reduced and it forms lactate. So NADH gets oxidized to NAD plus. Okay, so this is lactic acid fermentation. In the lactic acid fermentation, there is no carbon dioxide produced. So here it's very important to know that NADH, which is produced in glycolysis, NADH which is produced in glycolysis is converted. It's very important for it to get converted into NAD plus. So the recycling of NAD plus is very important, because otherwise the process of glycolysis will stop. Because in the process of glycolysis, NAD plus gets converted into NADH, but in cellular respiration, NADH delivers its electrons to the inner membrane of mitochondria, where those proteins are there, and then they eventually reach to oxygen. So in NADH delivers its electrons to the electron transport chain, and then NADH gets converted back to NAD plus, right? So similarly, it's very important for NADH to get converted back into NAD plus, because the job of NAD plus is to act as electron carrier. Its job is to bring more electrons. So if NADH doesn't get recycled to NAD plus, then the process of glycolysis will stop. So lactic acid fermentation usually takes place in our muscles. When we do like a vigorous exercise, you must have seen sometimes we feel pain, and it is because of the accumulation of lactate, because at that point when we are doing vigorous exercise, our cells need a lot of energy, and the oxygen supply is not that much. So then at that point the cells undergo lactic acid fermentation. Human muscle cells use lactic acid fermentation to generate ATP during strenuous exercise when oxygen is scarce. So because the amount of energy cell needs, the input of oxygen is not that much. So in that situation, the cells undergo lactic acid fermentation because it is quicker, but it gives less energy. Lactic acid fermentation is also used by some fungus and bacteria for making cheese and the yogurts. Okay. Okay, so this is all about the fermentation. So there is a yield of 2 molecules of ATP in the fermentation. These are some comparisons. You can go over them. It's just repetition. What are obligate anaerobes? Obligate anaerobes are the ones which carry out fermentation or anaerobic respiration, and they cannot survive in the presence of oxygen. So they are very, very, very strict with the anaerobic conditions. Facultative anaerobes are they can either undergo fermentation or cellular respiration according to the situation. So now you can see here, like this is glucose. Glucose gets converted into pyruvate. This is glycolysis. For glycolysis, oxygen may be there, oxygen may not be there. Doesn't matter. So, but after this step, if there is no oxygen, then fermentation will be the process of energy production. If oxygen is there, then aerobic respiration. So pyruvate will be converted to acetyl coenzyme A, then it will go to citric acid cycle and all that. Now something very important to know here is, in the beginning I said that in our food we eat different kinds of foods. Like in our food we have proteins, we have carbohydrates, we have fats. But when we study metabolism, when we study cellular respiration, we study by taking glucose molecule. We just start everything with a glucose molecule. But how do other molecules produce energy? So like fats, so you have already seen what are fats. They are glycerides. So they are basically converted first of all into fatty acids and glycerol can join this step here. If you remember in glycolysis, this is a step called glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is produced. So from the fats, when glycerol is produced, this glycerol gets converted into glyceraldehyde 3-phosphate, and it can join glycolysis here. And fatty acids, they can be converted into acetyl coenzyme A, and they can join here. So these are all some biochemical reactions. And then these proteins are first of all converted into amino acids, and then these amino acids they can be converted into pyruvate. Then pyruvate can join here, or they can be converted to acetyl coenzyme A, or they can be converted into some of the metabolites which are there in the citric acid cycle, and they can join the citric acid cycle. So this is the way other food products, food things which we eat in our food, they also give us energy, how they fall into this pathway and they give us energy. And the last thing I would like to tell you is about the regulation of cellular respiration, and it is by feedback mechanism. So basically, in our cells, it's not that our cells are producing energy all the time. So if the cells have a lot of energy already, then we have a feedback mechanism, and through that mechanism, the production of energy is stopped. And when the amount of energy is going down, the ATP levels are going down in the cell, then we have a mechanism by which the production of energy is increased. Now, if you look here, this is glycolysis. So glucose gets converted into glucose 6-phosphate, then glucose 6-phosphate gets converted into fructose 6-phosphate, and then fructose 6-phosphate gets converted into fructose 1,6-bisphosphate. Here is one enzyme involved. In the step from fructose 6-phosphate to fructose 1,6-bisphosphate, there's an enzyme involved called phosphofructokinase. So this enzyme is actually regulated. So when there is a lot of energy in the cell, then ATP, because when there is a lot of ATP in the cell, this ATP actually goes and inhibits this enzyme called phosphofructokinase. And when this enzyme is inhibited, all the steps further are going to be inhibited. And also the citrate from the citric acid cycle can also inhibit this enzyme, and the production of energy will stop. And when the ATP levels are less, then the cells will have more AMP levels. What is AMP? It's adenosine monophosphate. So adenosine monophosphate will be more, and then AMP can actually stimulate this enzyme. When this enzyme is going to be stimulated, the whole process down is going to be stimulated, and there will be more production of the energy. So that's all in this chapter. If you have any question, please let me know.